Very warm welcome to Dr. Najeeb lectures. Today we are going to talk about a very important topic, extremely important topic. The topic is bacterial protein synthesis, or we can call it prokaryotic protein synthesis. Why this topic is so important? Because there are so many antibiotics and drugs which act on the bacterial protein synthesis process and inhibit it or disrupt it, right? So it is very important to understand this process very clearly, to understand the mechanism of action of very important antibiotics. For example, these antibiotics, all of them mainly work on the bacterial protein synthesis and inhibit or disrupt the protein synthesis. For example, we can talk about aminoglycoside, linezolid. Linezolid is new drug and it is very important, especially for gram-positive multi-drug resistant bacteria. Linezolid, right? This also works on, especially is useful in methicillin resistant Staphylococcus aureus and even vancomycin resistant Staphylococcus aureus. But right now I will not go into detail of these antibiotics. I'm just trying to tell you that all these antibiotics are mainly disrupting or inhibiting bacterial protein synthesis, which include aminoglycoside, linezolid, chloramphenicol, macrolides. Macrolides, you must be knowing, erythromycin, clarithromycin, azithromycin, then clindamycin, tetracyclines, and another relatively new drug, TG-cycline, right? So all these drugs mainly work on the bacterial protein synthesis and disrupt it. So now we will discuss bacterial protein synthesis process in detail and step by step, right? No hurry, no worry. Once you understand it, slowly but properly, right? So that all your life you are not confused about the mechanism of action of these drugs. Now, when we talk about that how bacteria synthesize their proteins, basically that process is called translation, right? And this process has three main steps: initiation of protein synthesis, or we can say initiation of translation, elongation of translation process, and termination of translation process. It means that when we talk about bacterial or prokaryotic protein synthesis, we are going to discuss it in three steps: initiation process, then initiation process, then elongation of peptide chain process, and then termination of the translation process. Now, before I go really in detail, I would love that we should basically define what is meant by translation in genetic terms and molecular biology terms. What is meant by translation process? Translation is, you know, that protein synthesis process starts from the very first step is that from DNA we make RNA, right? Remember, DNA can be transcribed in messenger RNA and also into ribosomal RNA, also into transfer RNA and many other specialized small RNAs, right? Now, let's suppose this is DNA, right? Bacterial DNA. Now this DNA, first of all, the concept which I want to make it clear that this DNA, it can transcribe itself into messenger RNA. It can transcribe itself into, ribosomal RNAs from different genes, and it can transcribe itself into transfer RNAs, right? And even many other which I will not discuss now, like small nuclear RNA and many other RNA. What I want to tell you, that even though the genes which are present in bacterial DNA or even in eukaryotic DNA, here I make the DNA circular because bacterial DNA or bacterial chromosome is circular, right? Now, this part, this gene present over here, when this gene is transcribed, it makes messenger RNA. This is the messenger RNA. These are the codons or messenger RNA which are translated into peptide chain. Messenger RNA is the only RNA, right, where codons are translated into peptide chain, right? Ribosomal RNA help in formation of ribosomes, but nucleotide sequence in ribosomal RNA is not translated into any peptide chain, right? In the same way, transfer RNA does have many nucleotides, but those nucleotides or coding system is not translated into protein synthesis, and so forth about other specialized RNAs. What I mean by this, the very first thing I want to clear in your mind is that even though there are many different types of RNAs, the only RNA which is translated into peptide chain is messenger RNA, even though for translation purpose, ribosomal RNA and transfer RNA help, right? When we talk about translation process, as we will talk now, right, we will see there is a role of ribosomal RNA in translation, there is a role of transfer RNA in translation, but it is the messenger RNA, nucleotides or codon system which is translated into peptide. Now, how do we define translation to be more specific? We can say translation is a process in which, in simple way, translation is a process in which coding region of messenger RNA is decoded and translated into newly formed peptide chain. But we can refine this definition further. The simplest thing of translation is what? The process by which from messenger RNA we make peptide chain. That is a very rudimentary or primitive or very basic definition. But if you want to be more specific, what is translation exactly? You would say translation is a molecular process in which sequence of codons in messenger RNA are translated into sequence of amino acid in the newly formed peptide chain. Sequence of, what is translation? Sequence of, yes, codons in the coding region of messenger RNA are translated into sequence of amino acids in a newly formed peptide chain. But exactly how it happens, let's see that. First of all, in translation process, yes, only coding regions are translated. I will tell you later, there are some other parts of messenger RNA which are not coding for proteins, which are not coding for the amino acids. We will talk about that in few minutes. Another more elaborative definition will be translation is a molecular process in which molecular translators, what are molecular translators? Or messenger RNA translators. What are messenger RNA translators? They are ribosomes. So ribosomes are molecular machinery, macromolecular machineries, which are basically translator, which translate the codon sequence in the coding region of messenger RNA into amino acid sequence of newly formed peptide chain or polypeptide chain or nascent peptide chain. Now exactly see how messenger RNA work, how ribosomes work, and how transfer RNAs work in this process of translation. Let's first of all focus on the translators. The translators are actually macromolecules. What are those macromolecules? Ribosomes. Ribosomes. So ribosomes, let's talk about ribosomes first of all. What are ribosomes? Ribosomes are basically small macromolecular particles which are made of special type of RNA, which is ribosomal RNA. Ribosomes are made of ribosomal RNA and some proteins. They are basically very special aggregation of ribosomal RNA and ribosomal proteins, right? So now these RNAs, because they have RNA and they are having with it plus proteins, so we also call ribosomal particle ribonucleotide proteins. Ribonucleotide proteins. Now, when we talk about bacterial system, then there are two important types of ribosomes, right? There is a small subunit which is called 30S unit, and there is another subunit, right, which are 50S unit, right? There are two units of ribosomes in bacteria which are involved in translation process, that is 30S unit and 50S unit. 30S unit is also called small subunit of ribosome, and 50S is also called large subunit. Now, as 30S unit, if we go into detail of its composition, it will have ribosomal RNA. It will have ribosomal RNA, and ribosomal RNA which is present into it. Now this is RNA, yes, ribosomal RNA. This ribosomal RNA which is present in 30S ribosomal unit, this is called 16S ribosomal RNA. And along with that, there are about 20 proteins associated with it. For example, in more or less 20s. For example, in case of E. coli, there are 21 proteins. So we can say that small subunit of ribosome is called 30S subunit, and 30S subunit has 16S ribosomal RNA, right, aggregated with around 20 proteins. And why we use the word S over here, I will tell you in few minutes. In the same way, there is 50S ribosomal subunit, and when they are together, if they are separate, this is called 30S subunit, small subunit, and larger subunit, when it is separate, it is called 50S. But if they are together, then they are 30 plus 50 is not called 80S, it is called 70S ribosome. Again, let me explain. In bacteria, the ribosomes which are used to translate the messenger RNA into peptide chain, these ribosomes consist of basically two parts, smaller subunit and larger subunit. Smaller subunit is called 30S subunit and larger subunit is called 50S subunit, right? And if they are together, then we say they are 30 plus 50 does not make here 80. Here it makes 70S. What is the reason for that? The reason is that S unit stands for Swedberg unit. Swedberg unit, for example, we say it is 30S means 30 Swedberg unit. 50S means 50 Swedberg unit. These Swedberg unit are basically sedimentation coefficient, that when initially researchers put the cellular component into tubes and then they centrifuge it, they were looking at that how the cells organelles were sedimenting, and they were sedimenting at which speed, with what coefficient, right? And this depends on, sedimentation coefficient depends on not only on molecular mass but also depends on the shape, right? And when small subunit is alone, it is sedimenting relatively different rate. We say 30S subunit. When this is alone, it is sedimenting at the rate of 50 Swedbergs. But when they are together, right, they are sedimenting at the rate of what? 70 Swedbergs, right? So here I would like to a little reminder that this 30S and 50S, which make the 70S ribosomes, this structure is, this type of ribosomes are mainly present in bacteria, archaea, and even in mitochondria, because your mitochondria are actually derived, were evolutionary, they were probably once upon a time your mitochondria were bacteria, which eventually enter in your life and they entered into your cells, starting producing energy producing bacteria, and they are living in harmony with us, right? So mitochondrial ribosomes or bacterial ribosomes, both of them are actually consist of 30S and 50S subunit, and together we call them 70S subunits. But when we talk about eukaryotic cells, not prokaryotic cells, bacteria, they are not having a well-defined nucleus. They don't have any nuclear membrane, prokaryote. But eukaryotic cells, which are mostly our cells, right? Higher animal cells or higher life form cells, the eukaryotic cells which are having a well-defined nuclei, right? In eukaryotic cells, ribosomes are different from bacterial ribosomes. In eukaryotic cells, 30S unit in place of 30S unit in prokaryote. In eukaryote, it is 40S unit, right? In the same way, the larger unit in bacteria is 50S unit, and in eukaryote cells, it is 60S unit. And these together, they are called, yes, 70S ribosome. And if these two together, not 60 plus 40 are not called 100, but they are called 80S unit. So it means if someone is talking about 30S or 50S ribosomal units or talking about 70S ribosomes, he is referring to prokaryotic ribosomal system. But if someone is talking to you about 40S or 60S or combination of both of them, 80S ribosomal system, he is talking about ribosomes of eukaryotes. But right now we will focus on, prokaryotic system, right? That how in the bacteria ribosomes are working, right? Up to now we have only learned that bacteria has 30S subunit and they have 50S subunit, and these are the macromolecular machines which are responsible to translate the messenger RNA codons sequence into sequence of amino acid in the peptide chain. Now, initiation process. How the protein synthesis in bacteria is initiated, right? For this purpose, first of all, we will discuss about 30S unit in detail, right? 30S unit, now I'm going into detail. Now let's suppose here I'm not going to focus to 50S unit now, I'm only focusing on 30S unit right now. First thing, as I mentioned previously, 30S unit is basically, yes, made of which ribosome? Which ribosomal RNA? 16S ribosomal RNA. This S should be capital, right? Plus there are some proteins. These are ribosomal proteins, right? Now, a very important thing which is present over there is that what are the functional domains of 30S subunit, right? Especially which play a role in initiation of translation process. First of all, I will tell you that the 16S ribosomal RNA, this is 3' end, this here it has 3' end and here it has 5' end, right? Here near its 3' end, it has a very special type of sequence. I will go in detail of the sequence later, right? Very special type of nucleotide sequence, right? Which act as a sticky point, and this point help the ribosome to capture the messenger RNA to be translated, right? Because ribosome are going to read the messenger RNA, so ribosome should have some mechanism that messenger RNA should bind with it. So when messenger RNA of prokaryotic bacteria come, this is the point, right? Later I will go into detail of this nucleotide sequence. This is a unique nucleotide sequence, part of the 16S ribosomal RNA, which is responsible to stick on it or bind with messenger RNA so that messenger RNA properly bind and engage with 30S subunit and properly aligned here so that it can be translated. This is one point, right? Now, second point which I want to tell you about, that there are special soluble proteins, we call them initiation factors. These initiation factors help in the initiation of translation process. Now, out of these initiation factors, right, in case of bacteria, there's initiation factor 1, initiation factor 3, and initiation factor 2. 1, 2, 3. Okay, we can write it like 1, 2, and 3 initiation factor. And when you compare the process of initiation with eukaryotic system, in eukaryotic system, they also have their own initiation factors which are slightly different. And those initiation factors in eukaryotic are eukaryotic initiation factor number 1 and so on, so forth. There are many initiation factors involved in initiation of translation process in eukaryotic cells, and the special thing they have is that before IF, which is initiation factor, they are labeled with E. E means eukaryotic. So whenever somewhere it is written IF or initiation factor, if there is no E before it, it means these initiation factors are working for bacteria or mitochondria. But if before the initiation factor small e is written, it is denoting that these are the initiation factors which are used in translation process or in the eukaryotic cells, right? So these are eukaryotic, and the eukaryotic system has many initiation factor. Right now I'm not going into detail, I'm just making a little comparison. My main focus in this lecture will stay on bacterial protein synthesis or prokaryotic translation process. So what I'm saying, that this is our 30th subunit. Now before the translation process start, right, we must be having a very clear concept related with 30th subunit, and later on we'll talk about 50th subunit. Now in 30th subunit we have already discussed 16S ribosomal system. On its 3' end, it has a very special type of nucleotide sequence which help the prokaryotic messenger RNA to bind and stabilize and align properly on the 30th subunit, right? And there are some protein. Then I said there are special initiation factors. Now what are these initiation factors? What are these initiation factors doing over here? I will draw here initiation factor number 1 and 3. This is initiation factor number 1. Initiation factor number 1, initiation factor 1. It has a very special domain where it is binding. We'll talk in detail. And here is initiation factor number 3. Initiation factor number 3. Right? Initiation factor number 3. Here is the 3 initiation factor, and here is initiation factor number 1. Now what they are doing? They are binding with, yes, they are to start the process of initiation of translation. They are binding with 30th subunit at special areas. Now what are these special areas and what they are doing? They do many functions, but one of the function I will tell you is that, yes, they are okay. I will make it simple. They are raising their hands, right? They are raising their hands up. What they are doing? They are preventing the binding of 50th subunit to 30th subunit. Let me make it in a smaller way. This is 30th subunit. There are 3 special binding places. Then this is here. And here was your what? Now you have to tell me which subunit is here. Initiation factor number 1. And what is here? Here is your friend, initiation factor number 3, right? And they are having their special domain. And these special domains, you know what they are doing? They are not allowing, what is here? 50th subunit. They are not allowing 50th unit to approach the 30s. This is one function, right? So what we say, the role of initiation factor number 3 and initiation factor number 1 is to prevent the 50s subunit to bind with the 30s subunit prematurely. prevent the binding of 50F subunit to the 30F subunit before 30S unit is ready. Right? Now what I mean by ready? Before I go for that, I will tell you what are these things. These 3 cups. Now these 3 pockets are special pockets, right? These 3 pockets are special pockets which are transfer RNA binding pockets, or we call them transfer RNA binding domains of 30S subunit. We have these 3 pockets or 3 domains on the 30S subunit which are mainly concerned with binding of transfer RNA. Is that right? Now what is the function of transfer RNA? We just discussed that, right? Transfer RNA are basically, they are also made from the genes. There are many types of transfer RNA. Let me show. Suppose this is a transfer RNA in a very simple diagram. I show you this is transfer RNA, right? And what this transfer RNA is doing, that number 1, it has a special nucleotide sequence which will bind with messenger RNA nucleotide sequence. For example, this is messenger RNA and a trinucleotide sequence which is present on messenger RNA. This trinucleotide sequence which is signifying or relevant or coding for one specific amino acid, let's suppose lysine. Now this nucleotide sequence which is present on messenger RNA, and let us suppose it is coding for lysine amino acid, this will be called codon. Codon. And because peptide chain has multiple amino acids, so messenger RNA will also have multiple codons. These are nucleotide bases, right? And nucleotides here, and each triplet of nucleotide which is specifying here for a special type of amino acid, right? Coding for specific amino acid is called codon. For example, we say this is codon number 1 from here to here. I'm just giving an example. This is messenger RNA. Let's suppose here it is 5' end and here is 3' end. Now in this messenger RNA, here is codon number 1 and it is saying it is relevant with, it is coding for, suppose amino acid valine, right? And if it is doing for valine, now there should be one transfer RNA here which is carrying valine. This is a transfer RNA which is carrying here lysine, right? And so this codon is for lysine, this codon is for valine, and let's suppose this codon, codon 2 and codon 3. This codon, which is trinucleotide sequence in coding region of messenger RNA, let's suppose this is for, yes, or only for leucine amino acid. So there will be one more transfer RNA here which will be carrying leucine. Now, yes, you are. I will tell you. First of all, your question is very important, where all this is happening. Actually, this is the raw material for translation. Because for translation, we have to have a macromolecules like ribosomes. We have to translation coding system on messenger RNA. We have to have transfer RNA which can bring the amino acids and then there must be some mechanism by which amino acids are stitched together, or they are stitched or bound together to make a peptide chain. Now, bacterial cell already even before the translation start has many, what are these? Transfer RNA. And different transfer RNA are carrying different type of amino acids, right? So what are transfer RNA? Transfer RNAs are special type of RNA which are made by the genes. These genes are for the transfer RNA, and these transfer RNA come into the they are present in the cytoplasm or cytosol, and there every transfer RNA on its 3' end is able to bind with a special type of amino acid, right? And when transfer RNA is without amino acid, for example, this is transfer RNA, this has no amino acid, we say this is empty, it is not carrying, it is empty transfer RNA, or we call it deacylated transfer RNA, right? Or we say uncharged transfer RNA. You are right. Now there are special enzymes. These enzymes, right? These enzymes help to bind the amino acid with the transfer RNA. It means there are special type of enzymes present in the cytosol which are constantly binding different amino acids to specific type of transfer RNA. So it means they are loading the transfer RNA. They are making transfer RNA charged, right? Now, these amino acid loaded transfer RNA, they are called amino acyl transfer RNA. So what is amino acyl transfer RNA? Amino acyl transfer RNAs are those transfer RNA which are carrying their relevant amino acid. So amino acyl transfer RNAs, we can say loaded transfer RNA, we can say charged transfer RNA, right? Or amino acyl transfer RNA. During further discussion, if I use the word amino acyl transfer RNA, it means I'm referring to an transfer RNA which is loaded with its relevant amino acid. Now, I will not go into detail of the steps, but this enzyme which help to bind the amino acid with its relevant transfer RNA, this enzyme is making amino acyl transfer RNA. Because this enzyme is making or synthesizing what? Amino acyl transfer RNA. So this enzyme is called amino acyl synthetases. So this group of enzymes which can find specific type of amino acid and also specific type of what? Transfer RNA, and they utilize energy, and these enzymes attach them, right? So convert the simple transfer RNA, unloaded transfer RNA, or deacylated transfer RNA into loaded transfer RNA or charged transfer RNA or amino acyl transfer RNA. That is why these group of enzymes are called amino acyl transfer RNA synthetases. Is that right? Now what we know? For the process of translation, up to now, we are working on different domains of 30th subunit. We are still working on what is this? Initiation factors. And I also gave you some concept that codons are present on the coding regions of messenger RNA, and then there are transfer RNA carrying special type of amino acid, and these transfer RNA get loaded or attached by the relevant or specific amino acid with the help of amino acyl transfer RNA synthetases. Now, how it happen? Let's suppose this is amino, okay, I make it numerically. This is codon 1, 2, 3. and here is amino acid 1, 2, 3. Right. Now, if it is, this codon is having G, G, C, then which amino acid will fit here? The amino acid which can attach here should be complementary to it, and it should attach anti-parallel, right, and complementarily. So base pair here will be, if GCC is here, then there should be C, G and C hydrogen bonding here, G and C with G. Now they can bind with each other. This is called hydrogen base pairing. Now, what is happening? This sequence is codon, and its complementary sequence, which is present on transfer RNA, this sequence is called anticodon. It is called anticodon. So what is happening that, let's suppose this transfer RNA, it has uracil and guanine. Do you think it can bind here? No, because uracil will bind with adenine, not with guanine. This uracil, so it will only bind with adenine and cytosine. So what is happening? This codon is having this specific anticodon, right? And this codon number 2 will only bind with other transfer RNA, and codon number 1 cannot bind with transfer RNA with anticodon number 2. It will bind with only its complementary. So what really happens, that when messenger RNA, we will see later, when messenger RNA, right, will be present on the ribosome, right, transfer RNA will bind there only when there, what is there? Transfer RNA and messenger RNA codon anticodon system is complementary and anti-parallel, right? Complementary, you understand that, for example, guanine with cytosine, or cytosine with guanine, or adenine with uracil, right? This is complementary system. Anti-parallel mean that if this is 5 to 3, this is 5 and to 3 end, then if codon is, okay, I can put it here, if codon is 5 to 3, here is 5 end and 3, then anticodon has to be anti-parallel and complementary. Complementary mean refers to the base pairing, anti-parallel mean it should be 3 to 5, right? So we say that there should be matching between the codons and anticodons in such a way that they are complementary to each other and they are in anti-parallel orientation with each other. Then a specific codon will be binding with specific anticodon of transfer RNA, which will be carrying a specific amino acid, right? This is the basic principle, that how codons are translated into bind with anticodons and eventually refer to or translate into presence of a specific amino acid, right? Now let's go back. I was just telling you some basic principles, right? Let's go back. Now what really happens? For the initiation process, as I told you, we needed, what was this? 30th subunit. We needed initiation factor 1 and initiation factor 3, especially initiation factor 3, which both of them and more of this is responsible to prevent the 50th S unit to bind with 30th unit prematurely. Because before 50S subunit before 50s subunit bind with 30s, there are some more molecular steps to be taken. There are few molecular steps to be completed by 30s system. Now, first step is, I purposely have not talked about initiation factor number 2. But let me talk about these pockets, right? These pockets. Again, I will come to I will talk about these other functional domain of 30s subunit, that let's suppose of course you know that these were the, what was these areas for, yes my friends, initiation factors, binding areas, right? Now what are these 3 pockets? What is this pocket? What is this pocket? And what is this pocket? These 3 domains. Of course 16 S is present over here, and here was special point which will bind the messenger RNA. What are these pockets? Actually, these pockets bind the transfer RNA. These 3 pockets are transfer RNA binders, right? Which we will see later. But when transfer RNA are binding here, they arrive at this point. So this pocket is called arrival pocket, or we simply call it new transfer RNA. Suppose this is a transfer RNA which is coming here with an amino acid. So because it is loaded with amino acid, so it is amino acyl transfer RNA. Now, first pocket here, this will bind the incoming or newly arriving transfer RNA, right? So this pocket or binding point or domain of 30th subunit is called A point, A site. On 30S, there is A site. But here I want to specify specially that this A site is not only present, okay, this is later on, I will show you when 50S bind here later on, not now. Actually, A site is partially on 30S and partially on 50S. So all this is A site. A site of 30S, A site in the 50S. What is A site? This is for arrival of, A for arrival of, yes, arrival of, what is this? Amino acyl transfer RNA. Amino acyl transfer RNA. So actually, we call this A site because of amino acyl transfer RNA binding site. Most of the time, this is for the arrival of newly coming amino acyl transfer RNA, right? Then the second pocket, this also partially extend on 30S and partially extend on 50S subunit, right? Slightly on 30S and large component on 50S. Here, the amino acid which initiate the peptide formation, or transfer RNA which initiate at this point. What is here? Here is a transfer RNA binding site. Which transfer RNA? Either the transfer RNA which is the first one in initiating the peptide chain formation, or when peptide chain is being formed, that transfer RNA which is having the newly formed peptide chain, we'll see it later. The transfer RNA which is having, for example, this is transfer RNA here, right? And it is having what is this? Newly formed peptide chain, right? This transfer RNA, which is having few amino acids already, because peptide chain is being synthesized, as we will see later, this transfer RNA is called peptidyl transfer RNA. Now, what is the difference between, this is coming here, what is the difference between this transfer RNA? Yes, this has one amino acid only, this is called, but incoming transfer RNA, this is called amino acyl transfer RNA, and this is called A site. Adjacent to that, there is another binding site where those transfer RNA bind, which are either carrying the initiator amino acid, which we will discuss later, or they are carrying the newly formed peptide chain. So, because this transfer RNA, which is carrying the newly formed peptide chain, so this transfer RNA is called peptidyl transfer RNA, and this site is called P site. This is called P site. If this was A site for amino acyl transfer RNA arrival, so this is P site. Why we call it P site? Because here is peptidyl transfer RNA binding. For us, we can also remember this is the parking site. Here is the arrival site. Here is the parking site where amino acid is either initiator, transfer RNA is there, or such transfer RNA is there which has newly peptide chain. And then we come over here. Here are usually, here was amino acyl transfer RNA binding, amino acyl transfer RNA. Here was transfer RNA with newly formed chain, right? And here is the transfer RNA which has given its amino acid to the peptide chain, and now it is about to leave. This was coming here, this is going to exit. Because this is going to exit, right? We call it E site. What we call it? E site. So, what we see there? In the 30S ribosome, what we see there, that here were three special transfer RNA binding domain, right? One domain, then other domain, then another domain, right? Now, this was called A site because it welcomes the upcoming or newly coming amino acyl transfer RNA. Then there is P site. This was arrival site. This is parking site, where such transfer RNA is present. Either it is going to initiate the process of translation, or it is that which is carrying a peptide chain. So P site. And here transfer RNA I have shown with one amino acid, right? Here is transfer RNA with newly formed, yes, peptide chain, peptidyl transfer RNA. What is it? Peptidyl transfer RNA. And here is the transfer RNA which has already given its, what is this? Amino acid. It is no more amino acyl transfer RNA, it is no more peptidyl transfer RNA, it is decylated transfer RNA or empty transfer RNA. So, A site is for amino acyl transfer RNA which receives the new, and P is parking place where transfer RNA with newly formed peptide chain or initiator transfer RNA is there, and this is for the exit point. So, A site for amino acyl, P for peptidyl transfer RNA, and E for from where that transfer RNA is present which is about to exit the ribosomal system, right? Now, how many domains of 30S subunit we have discussed?